Hello class, I've got an exciting first day plan for you all. But before we do that, let's go over what we learned last class. Specifically, I want you guys to tell me about cell theory.
Teacher, cell theory says that cells come from other cells.
Why yes, that concept is known as biogenesis.
Teacher-son, cell theory also has it that all living things are made of cells.
And it also says that cells are the basic unit of structure and form. Very good, class. Today we're going to travel into a cell and see the organelles firsthand.
What?
He can't be serious.
This teacher's a phony. This is of course an intimidating task, considering eukaryotes are usually 10 to 100 micrometers in size. In order to facilitate entry into the cell, we shall be using the magic unmarked white van. Come on everyone, inside. Whoop 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 whoop. Ain't the cell beautiful? It's only at this size that we can truly appreciate it. It's a shame. In order to get into this cell and view its internal components, or as we know them, organelles, we must first pass through the cell membrane. Some people might call this the plasma membrane. It's a semi-permeable membrane composed primarily of a phospholipid bilayer. These phospholipids have a hydrophilic head and a hydrophobic tail. The cell membrane is also composed of proteins. These proteins can be used as either signals to other cells, receptors for incoming matter. As we know, cells can either take in materials through passive transport or active transport. Passive transport would require no energy and transports materials from high concentration to low concentration, whereas active transport would transport them against their concentration gradients. That is to say, from low concentration to high concentration. What we are standing in now is known as cytoplasm. This gelatinous goop is primarily water with some proteins and holds all of the cell's components outside of the nucleus. If you look further onwards, the large and most visible part of the cell can be seen. This is the aforementioned nucleus. It houses the cell's genes and is the control center for all cell activities. It is enclosed in a nuclear envelope separating it from the cytoplasm. Pores along the envelope regulate the entry and exit of molecules from the nucleus. Before we head that way, let's take a look at the other organelles of this cell. Here we can see a centriole. This is a cylindrically shaped cell structure, which is composed of 9 triplets of microtubules. These are absent in plant cells, but in animal cells, they are used during cell division to pull the cell apart.
What's that, teacher?
Here, we have some microtubules, also known as tubulin polymers. These are one of the components of the cytoskeleton, which can support the cell, produce motility, and move vesicles. Microtubules are hollow tubes with a diameter of 25 nanometers. These polymers can additionally move chromosomes during cell division. And here we have microfilaments, also known as actin filaments. These are the thinnest filaments of the cytoskeleton. These measure approximately 7 nanometers in diameter. Ribosomes can either be free in the cytoplasm or bound to the rough ER. Free ribosomes make proteins for use inside the cell, while bound ribosomes make proteins for export. The ribosome consists of 2 subunits and translate RNA into proteins. This, of course, is a bound ribosome. Here we begin moving through the endoplasmic reticulum, the outer layers of which are called the smooth endoplasmic reticulum. This is able to synthesize lipids, metabolize carbohydrates, detoxify poisons, and store calcium. Furthermore, if we look at the rough ER, this section has bound ribosomes, which secrete glycoproteins. These proteins are covalently bonded to carbohydrates. And here, we can see the Golgi apparatus.
Hehehe, that's a funny name.
Why yes, it was named after an Italian scientist who discovered it. This organelle can be described as a shipping and receiving center. As you can see, it consists of a flattened membranous sacs called cisternae. The Golgi apparatus is able to modify products of the endoplasmic reticulum, manufacture certain macromolecules, and sort and package materials into transport vesicles. And here we have a vacuole. These are storage organelles and are much smaller in animals than they are in plants. And this is a vesicle. These are small bubbles within a cell enclosed in a lipid bilayer. They are the basic tool used for organizing cell substances and can be used for transport of materials. Coming into focus is a mitochondria. Crucial to the ATP cycle, the mitochondria is the site of cell respiration. Mitochondria have a double membrane, have proteins made by 3 ribosomes, and even contain their own DNA. Mitochondria are not part of the endomembrane system. And here we have cilia, coming from the Latin for eyelash. It's an organelle which is found in eukaryotic cells and is used for either movement or sensing.
What's that, teacher?
That's a lysosome. Lysosomes are cellular organelles that contain acid hydrolase enzymes. They break down waste materials and digest cellular debris. And now they're gonna digest us. Oh my god! Quick, everyone into the unmarked white van. We must evade. Quickly, we need to get to the nucleus. The nuclear envelope will protect us. Quick, evasive maneuvers. Dive, dive, dive. It's gaining on us. Oh no! We're almost to the nucleus. Just a bit farther. Phew, that was a close one. I hope I can speak for everyone when I say my heart was racing during that chase segment. That might have been even too intense. But alas, we are now safe within the envelope of the nucleus.
Ew! What's all this genetic material?
That's chromatin, an uncondensed form of DNA composed of nucleotides and protein.
And what's that big thing over there?
That's the nucleolus. It sits within the nucleus. What was that? It sounded like the very core of the nucleus was being destroyed. Oh no, the cell is undergoing mitosis. Soon, the nucleus will break apart, and then we'll be defenseless to the lysosome. What will we do? Oh no!